Chapter 14 is where we initiate our discussion on metabolic pathways, starting with glycolysis. Glycolysis, as you may know, involves converting glucose to pyruvate, a metabolite that is involved in energy production. This chapter involves individual reactions of glycolysis, gluconeogenesis, which is the reverse of glycolysis, and pentose phosphate pathway. In addition to this chapter will also discuss the functional significance of each of these pathways. We will also discuss about various metabolic fates of pyruvate, which is the product of glycolysis. This lecture involves our discussion on glycolysis. To be specific, the first part of glycolysis, the preparatory phase, is discussed in this chapter. The learning goals of this chapter involve harnessing energy from glucose via glycolysis, fermentation under anaerobic conditions, synthesis of glucose from simpler compounds, that is called as gluconeogenesis, oxidation of glucose in pentose phosphate pathway, an alternate pathway to glycolysis. So the obvious question is, why glycolysis? And that has to do with the central importance of glucose. Glucose, as you may know, is an excellent fuel. One of the reasons is because it yields good amount of energy upon oxidation, about 2,840 kilojoules per moles per glucose molecule. Now, in addition, it can be efficiently stored in polymeric form. If you remember, glucose is indeed stored as glycogen in our body. Many organisms and tissue can meet their energy needs on glucose only, and hence glucose is an excellent fuel. The second reason is that glucose is a versatile biochemical precursor. Many organisms can use glucose to generate other molecules, like all the amino acids, membrane lipids, nucleotides in DNA and RNA, and cofactors needed for metabolism. And hence, glucose has a very important place as a central metabolite. Discussed here are the four major pathways of glucose utilization. The first pathway is storage. Glucose can be stored in the polymeric form, either starch or glycogen, and it can be used for energy needs later. It can be used for energy production, generates energy via oxidation of glucose. As I told you before, complete oxidation of glucose can give about 2,840 kilojoules per moles of energy. It is used for short-term energy needs. For example, if you want to take a sprint, you take glucose and you get that extra burst of energy to just sprint down. Production of NADPH and pentoses generates NADPH, which is a reducing agent in our body for use in relieving oxidative stress and synthesizing fatty acids. Those are the two uses of this molecule, NADPH. Remember, NADPH is just a cousin of NADH, right? It's a reducing agent and it is used to relieve oxidative stress in our body and also as a cofactor in synthesizing various fatty acids. In addition, glucose is also used in generating pentose phosphates for use in DNA and RNA biosynthesis. Finally, structural carbohydrate production. It is used for generation of alternate carbohydrates used in cell walls of bacteria, fungi, and plants. And also those various oligosaccharides that are decorated on proteins outside the cells. If you remember carbohydrates chapter, we learned about proteoglycans and glycoconjugates. Now, all those sugars that we learned about can be synthesized from glucose itself. Glycolysis literally means breaking down glucose, and in this case, breaking down glucose to pyruvate. This provides energy for cells through formation of ATP and NADH. In this process, called as glycolysis, glucose gets converted to 2 molecules of pyruvate. And it involves 10 successive reactions. And while doing so, this process generates ATP and NADH, which are sources of energy. Glycolysis can be done in 2 ways. One is via anaerobic fermentation pathway without oxygen, or via aerobic pathway, which involves oxygen. So glycolysis, where glucose is converted all the way to oxygen, can be via aerobic conditions or via anaerobic conditions. Now, aerobic glycolysis, which is shown here, provides significantly more energy as compared to anaerobic hydrolysis or glycolysis. Before we discuss the various reactions involved in glycolysis, it is good to know the importance of this central metabolic pathway. Research on glycolysis played a large role in the development of modern biochemistry, such as understanding the role of coenzymes, discovery of the pivotal role of ATP, development of methods for enzyme purification, and last but not least, inspiration for the next generation of biochemists. And because of this, biochemistry students have been forced to learn about these series of reactions for decades. And if I'm right, decades more to come. Here is a quick overview on glycolysis. In the evolution of life, glycolysis probably was one of the earliest energy-yielding pathways. It developed before photosynthesis, when the atmosphere was still anaerobic. Thus, the task upon early organisms was how to extract free energy from glucose anaerobically, or in the absence of oxygen. The first solution, obviously, was to activate glucose by phosphorylation. Second, collect energy from the high-energy metabolites. So this is very important. Glycolysis is a 2-stage process. It has a preparatory phase and a payoff phase, which means you're investing something in this phase and you're getting in return in the next phase. The preparatory phase breaks glucose down into 2 molecules of glyceraldehyde 3-phosphate molecules. So, in this phase, net input of free energy, which means that the cell is investing in the first phase because it knows that that phase is going to provide with a molecule or with 2 molecules that will give the cell in return more energy than it invested. The payoff phase converts each glyceraldehyde 3-phosphate molecules into pyruvate molecules. So, net yield of free energy. So the point here is that in converting glucose to two molecules of glyceraldehyde 3-phosphate, the cell invests some energy. And these two glyceraldehyde 3-phosphate molecules are separately converted to two pyruvate molecules. And this provides more energy than what is invested. And hence, the difference between the invested and the yielded energy is your return. And that is how glycolysis makes energy. Let us consider the first phase in glycolysis. It is the preparatory phase. And in this phase, glucose gets converted to glyceraldehyde 3-phosphate, two molecules of them. And if you look closely, there are two priming reactions, the first and the second. The first priming reaction is when the cell invests an ATP molecule, and the second priming reaction, another ATP molecule. So, two ATP molecules are invested, and hence it's the preparatory phase. The second phase of glycolysis involves the payoff phase. This involves two ATP forming reactions. And because there are two glyceraldehyde molecules, the cell gets 2 + 2, 4 ATP molecules in total from this phase. Besides, it also gets 2 NADH molecules. Now, it is important to remember that one NADH molecule is equivalent to 2.5 ATP molecules. So, in total, the cell invests 2 ATP molecules in the preparatory phase, and it gets back 4 ATP molecules in the payoff phase and 2 NADH molecules. So, if you total it together, the net profit is 2 ATP molecules plus 2 NADH molecules. Now, because NADH is equivalent to 2.5 ATP molecules, 2 NADH becomes 5 ATP molecules. So 5 + 2, 7 ATP molecules is the net yield in converting one glucose molecule to two pyruvate molecules via glycolysis. Let us look at step 1 of glycolysis. Step 1 involves phosphorylation of glucose. So this is glucose, and it gets phosphorylated to glucose 6-phosphate. Rationale for this reaction is that glucose gets trapped inside the cell. Again, why? Because phosphorylated molecules cannot escape cells, nor can they enter cells without specific transporters. Glucose, on the other hand, can get out of the cell or get into the cells. So, phosphorylation of glucose, by phosphorylating glucose, the cell makes sure that glucose is trapped inside the cell. Second rationale is lowers intracellular glucose concentration to allow further uptake, because the cell realizes that there is less glucose inside the cell, more and more glucose is taken up. This process uses the energy of ATP. If you look closely, ATP gets converted to ADP. And this is catalyzed by the enzyme hexokinase. There are multiple isoforms of hexokinase that exist in organisms. Example, hexokinase 1, 2, 3, and 4. These are all glucokinases. Now, if you consider this reaction, the mechanism is pretty straightforward. The nucleophilic oxygen at C6 of glucose attacks the last phosphate of ATP, and ADP is released and the phosphate is attached to the C6 hydroxy of glucose to form glucose 6-phosphate. The ATP is bound to magnesium, and that facilitates the shielding of negative charges on ATP. This is a highly thermodynamically favorable and irreversible reaction, as seen from the standard free energy change, which is about minus 16.7 kilojoules per mole. And the total free energy change, or the actual free energy change, which involves the concentration of glucose 6-phosphate and glucose, also is favorable because glucose 6-phosphate is utilized in further reactions. And it is regulated mainly by substrate inhibition. Step 2 of glycolysis. This step involves the conversion of glucose 6-phosphate to fructose 6-phosphate. And this reaction is an isomerization reaction. This reaction is catalyzed by this enzyme Phosphohexose Isomerase, which uses magnesium as a cofactor. The rationale for this reaction is that making next step is easier because less energy is required. C1 of fructose, which is this carbon, is easier to phosphorylate by phosphofructokinase, allows for symmetrical cleavage by aldolase. Now, these two rationale becomes clear once we look at the next three reactions. Now, you may know that an aldose, which is glucose 6-phosphate, can isomerize into ketose, in this case, fructose 6-phosphate. What you know from what you learned in chapter 6 is that glucose is also an aldose that gets isomerized to fructose, which is a ketose. And it goes through an enediol intermediate. This reaction is slightly thermodynamically unfavorable and hence slightly reversible. The product concentration is kept low by pairing with favorable next step to drive reaction forward. So what I mean to say that even though the standard free energy change is positive, the actual free energy of the reaction is negative because fructose 6-phosphate concentration is kept low in the cells. So the reaction quotient Q becomes less than 1, and hence delta G, the total actual free energy change, becomes negative. The mechanism of phosphohexose isomerase is what we looked at in chapter 13. If you remember, we briefly looked at this phosphohexose isomerase reaction. And let me introduce this mechanism one more time. The first step is the binding of glucose 6-phosphate to the active site of phosphohexose isomerase, and once it binds, the six-membered ring opens up to a linear aldehyde conformation. And the enzyme has a glutamate in its active site that acts as a general base and an acid. And it abstracts this proton, which is acidic, and it pushes the bond between C2 and C1, and the aldehyde carbonyl is now polarized and takes up a proton. And proton abstraction from this glutamate gives rise to this kind of an enediol intermediate. This is a cis-enediol intermediate. And the base, which has now contributed the proton to this hydroxy, takes up this proton, and the bond is now pushed in to between the carbon and oxygen, and the bond between C1 and C2 now takes up a proton from the original base, which is now protonated. Now, as a result, you get fructose 6-phosphate in a linear conformation, which now closes to form a cyclic fructose 6-phosphate. Step 3 of glycolysis. Step 3 of glycolysis involves conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. If you look at the structure, the hydroxy on carbon 1 of fructose 6-phosphate is phosphorylated by the enzyme phosphofructokinase 1, which uses ATP as a cofactor. The rationale for this reaction is that it further allows for activation of glucose. It also allows for 1-phosphate or 3-carbon sugar formation after step 4. Most importantly, this is the first committed step of glycolysis, and this reaction is catalyzed by phosphofructokinase 1. Fructose 1,6-bisphosphate, the product of this reaction, is committed to become pyruvate and yield energy. Furthermore, this process uses energy of ATP. This is the second investing step. This is highly thermodynamically favorable, again, because ATP is used in this process. Any reaction which involves ATP is a thermodynamically favorable process because the energy involved in hydrolysis of ATP is coupled in this reaction. Phosphofructokinase 1, the enzyme that catalyzes this reaction, is highly regulated by, first, ATP, second, fructose 2,6-bisphosphate. This is an intermediate that we have not seen. But this is an important intermediate because this intermediate activates phosphofructokinase 1. There are other metabolites that also regulates phosphofructokinase 1. But most importantly, fructose 2,6-bisphosphate. The presence of this metabolite is what initiates the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Step 4 of glycolysis. Step 4 involves conversion of fructose 1,6-bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And this reaction is catalyzed by the enzyme aldolase. And this involves aldol cleavage of fructose 1,6-bisphosphate. The rationale for this reaction is that the 6-carbon sugar, which is fructose 1,6-bisphosphate, is cleaved into 2 3-carbon sugars. High energy phosphate sugars are indeed 3-carbon sugars. The reverse process is the familiar aldol condensation. Multiple mechanisms exist to result in the same product. For example, animal and plant aldolases employ covalent catalysis in their reaction to convert fructose 1,6-bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, whereas fungal and bacterial aldolases employ metal ion catalysis. Now, if you notice, this is a thermodynamically unfavorable process, breaking of fructose 1,6-bisphosphate to 2 3-carbon phosphate containing sugars. The product, which is glyceraldehyde 3-phosphate, concentration is kept low to pull the reaction forward, and hence the actual free energy of the reaction is negative. The mechanism of class 1 aldolases, or aldolases in animals and plants, is shown here, and it goes through a covalent mechanism. Let me take you through individual steps, one by one. In the first step, fructose 1,6-bisphosphate binds to the enzyme aldolase, and as a result, ring opens. And the next step is the attack of a lysine. Now the lysine has an NH2. Now this attacks this carbonyl, and this is like a Schiff's base reaction. If you know what a Schiff's base is the reaction between amine and an aldehyde or a ketone to form an imine. Now the way it happens is that the lone pair attacks the carbonyl center, the carbonyl carbon, and the carbonyl bond polarizes to abstract a proton from an active site acid. Now, as a result of this, a base abstracts the excess proton on the NH, and it pushes the bond and pushes out the hydroxy. This results in a protonated Schiff's base formation. Now, Schiff's base, the reason why a Schiff's base forms is because a Schiff's base is a good electron sink, which means that electrons can be pushed into that nitrogen, which is positively charged. Now, as a result of this, the hydroxy proton can be taken up by another base, and it can push the electron into this carbon-oxygen bond and cleave a carbon-carbon bond. That is a big process. That is exactly why a Schiff's base is formed, is because one can actually cleave this carbon-carbon bond. You can push this bond to the next carbon here between carbon 2 and carbon 3. So the bond between carbon 3 and carbon 4 is broken, and the electrons are pushed in between carbon 3 and carbon 2, and as a result, the Schiff's bond, the double bond, can be pushed into the nitrogen, which is now an electron sink. So this is the key reaction that happens in this process. Now, once this happens, there are two different molecules. A glyceraldehyde 3-phosphate is released as a result of this cleavage, and what is stuck to the enzyme is this lysine adduct. Now, in the next reaction, because the lysine is now deprotonated, the lone pair on this nitrogen moves, and the double bond that is formed between C2 and C3 now takes up a proton from an amino acid in the active site of the enzyme. As a result of which, there is an isomerization reaction. Now, the isomerization reaction, as you can see, is what I just indicated. And as a result, what you get is this molecule here. Now, once that molecule, the protonated Schiff's base, is formed, it can release itself because water can attack the Schiff's base, and it can release the lysine because the reaction is complete. And once this lysine is released from the molecule or the product, which is dihydroxyacetone phosphate, is released from the enzyme. So the complete reaction is conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, which is released first from the enzyme, and dihydroxyacetone phosphate, which is released next as the next product. Step 5 of glycolysis. Step 5 involves conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, catalyzed by the enzyme Triose phosphate isomerase. The rationale for this process is that it allows glycolysis to proceed to payoff phase by a single chemical pathway. So, this reaction makes sure that there are two glyceraldehyde phosphate that are now entering the payoff phase. Aldolase creates two triose phosphates, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And dihydroxyacetone phosphate now gets converted to glyceraldehyde 3-phosphate by triose phosphate isomerase, and hence 2-glyceraldehyde 3-phosphate are produced. Only glyceraldehyde 3-phosphate is the substrate for the next enzyme, and hence dihydroxyacetone phosphate must be converted to glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase so that the payoff phase can begin. Now this completes preparatory phase of glycolysis. Thermodynamically unfavorable. As you can see, this reaction has a standard free energy change of 7.5 kilojoules per mole. The way this reaction proceeds is by keeping the glyceraldehyde 3-phosphate concentration low, which means that when glyceraldehyde 3-phosphate is formed, it is used up for the next reaction. And hence the actual free energy change for this reaction is negative. So the preparatory phase is now complete. Total effects is that one molecule of glucose gets converted to 2 molecules of glyceraldehyde 3-phosphate. 2 ATP molecules are invested in this step by the cell to make 2 ADP molecules. So the overall delta G0 for the step is a combination of all the standard free energy change for individual steps, starting from step 1, step 2, step 3, step 4, step 5. If you add all these, the total free energy change is 2.1 kilojoules per moles. But the way cells maintain a negative free energy change for the actual free energy is by pulling the products further down. And because the product of the reaction is glyceraldehyde 3-phosphate and these molecules are used up instantaneously in the next reaction, the preparatory phase goes forward to the payoff phase.